Welcome everybody to this webinar. It has a very special title for very special indications of gene therapy. So the title is N=1, the gene therapy development for ultra-rare disorders. This is part of the series of webinars we organize from the Gene Therapy Working Group, which is an European Reference Network working group where the ERN rare neurological, ERN rare neuromuscular, and ERN epicare collaborate to promote awareness and collaboration and education on gene therapy applications, which is becoming, of course, more and more important in our research and clinical practice. And so today I'm very happy to welcome Dr. Terry Pirovolakis as our speaker. Terry is really a very appropriate speaker for this topic because he has gone the path of N=1 gene therapy development, and you will hear his story. Terry is a co-founder and CEO of Cure SPG50, and also he established Elpida Therapeutics. His goal is to treat very rare and ultra-rare neurological diseases. He has done groundbreaking research treating 3 children within the last 3 years. And through the establishment of Elpida Therapeutics, he also collaborates with non-profit organizations to develop also therapies for other ultra-rare conditions. Currently, Elpida Therapeutics also targets, in addition to SPG50, CMT4J, and also has some other disorders on the pipeline, all of which are ultra-rare. And Dr. Pirovolakis also supports foundations that providing gene therapy 101 classes, engaging in collaborations, all centered on the goal of saving as many children as possible. So also seen as a matter of urgency to develop treatments for these patients. So Terry, thank you very much for accepting to talk. So I will give you the floor to share your slides. And then after the end of the talk, we will have the question and answer session. So I would like to ask the audience if they have questions or anything to discuss, to please note it down in the chat or in the Q&A window, and then we can discuss it after the end of the talk. Thank you very much. And Terry, the floor is yours.
Thank you very much, Doctor. Can you see my screen? Everything's okay? Perfect.
Yes. Yeah.
Just one thing to clarify, I'm not a doctor. I'm just a regular guy. Yeah, I've done enough work recently, that's for sure. So my name is Terry Pirovolakis. The last 25 years, I've been an IT director for large banks across Canada. Most of my work was in Latin America, in the Caribbean, where I did large deployments of infrastructure for technology. We did AI models, we did CRMs. It was just very large implementations. And on December, sorry, I apologize. This is my family. My name is Terry. My wife is Georgia. My oldest daughter is Zoe. My middle son is Zach, and the grumpy baby is Michael. My son was born December 17, 2017. We left the hospital. It was a normal, healthy birth, nothing out of the ordinary. We were home within 3 hours, being our 3rd child. We then noticed around 5 months of age that Michael wasn't lifting his head. He wasn't lifting his hands, and he was in the 10th percentile at that point for head growth. But the doctor said, don't worry, go home. Things should catch up. He'll be perfectly fine. And we continued on with our lives. Around 6 months of age, we reached out to the pediatrician because even though he was making gains, as you see, he was reaching for things. He still wasn't reaching the milestones that he should be for the age. He had low muscle tone at this point in time, diagnosed. and he was at the 5th percentile for head growth in the World Health Organization charts. But as you see, he began reaching for things. We began intense physiotherapy, and we were moving along. At 8 months of age, we noticed, again, he still wasn't reaching his milestones. So they sent him to, we were sent to infectious disease center at Sick Kids in Toronto. And the reason for that is they thought he contracted Zika because that was in Latin America. At the time, Zika was a big thing. And then at that, they ruled out Zika. They then thought he had CMV. We ruled that out. We did all the panels. And then we ruled out all the infectious diseases. At this point in time, he still wasn't reaching his milestones. He was at the 3rd percentile for head growth. Sorry, I'm a bit sick this past couple of days. And his tone was improving. As you can see, he was sitting up in this little box, but he still wasn't making the gains that he should be. And then at 10 months of age, all the infectious diseases were ruled out. And this point in time, they were telling us that it was becoming serious. So we took a pilgrimage to Greece. This is me and my wife and Michael in Greece, and we to pray for Michael's disease to go away. But unfortunately, this is something we couldn't pray away. We were sent to neurology, and we noticed at this point in time he was diagnosed with severe microcephaly. His MRI came back showing that he had thinning of the corpus callosum, abnormal white matter, but his tone was improving and he was making gains. At 12 months of age, they ruled out all the panels at this point in time. They collected a sample for a West report, a whole genome sequence report, and he was in the -3 percentile for head growth. As you see, he began to crawl, he began to pull to stand, and again was still improving. And then on April 2nd in 2019, he was diagnosed with this terrible disease called spastic paraplegia 50 through a West report done at the hospital. They told us, go home, love him. There's nothing much that we know about the disease, not much we can do. We do know he'll be paralyzed from the waist down by the age of 10. He'll be quadriplegic by the time he's 20. He'll have severe developmental delays. He most likely will never speak. He will never walk. And we don't know about his lifespan. We believe it's limited. We were just devastated. We went home crying. It was a fog for basically 2 days. And me and my wife said we have to do something. We have to try to save Michael. And we started researching online that evening. We found a family in Boston who started talking about gene therapy and ASOs and drug screening, and we had no idea what any of this meant. But we knew that there was something that could be done. There was hope there. And that kind of led us down this journey of kind of doing, like what we said we were going to do, which is we were told saving Michael was impossible, but for us, impossible was doing nothing. So I flew basically 1 month to the day after Michael was diagnosed to the ASGCT conference in at the time it was in Washington, and I met 6 of the 7 world experts in gene therapy. I then flew to Sheffield, England. I met the 7th world expert in gene therapy at the time, anyways. And then I went to the FDA, the NIH, and I attended a boot camp at Ultragenics to learn how to make a gene therapy for my son and the other children that were affected. At this point in time, we had signed a research agreement. We had collaborated with the NIH to do testing on fibroblasts. We supported a natural history study at Boston Children's. We collaborated to test the gene therapy in Boston Children's as well. We funded iPSC cell lines at Boston Children's. And unfortunately, we had to reverse mortgage our home and spent our life savings to start the process of making the gene therapy. And again, we met Dr. Stephen Gray and Dr. Xin Shen at that conference, and I hired them and they're in the hospital, or partnered, that's a better word, to make the gene therapy for my son and all the other children that were affected by this terrible disease. And at that point in time, we knew we had to raise $3 million, which was a daunting task. And we came out with our to our community, and we basically said, we have to raise $3 million. Michael has this terrible disease. We don't know if it's going to work. We don't know anything at this point in time, but we do know we have to try. And the 2 weeks later, they did a Beerworks event, as you see on the bottom right. They made a beer under the charity that we raised, or we created, and we raised $25,000, and that was kind of the beginning of our journey to raise the money and to save Michael. And then the person on the left, his name is Bottle Bob. And this amazing guy goes around the community, collects pop cans and bottles, recycles them for money. And then he was like, I'm going to come every month with money to help you guys out. And every month he does. It's pretty amazing. He donates all the money. He just wants to help the community. And then this is a short video, but basically this was a catalyst for us to go viral and help us raise the money that we had to do. But I'll play the video right now, and hopefully you can hear it.
Hi, baby.
We're going to cure him. That's what we're going to do. We're going to cure him.
Or give him the best shot.
Look at all that stuff to play with. Should have recorded that. So tell me, what's Michael like?
He's a pretty happy kid in general. Michael's a happy little guy. You know, he works really hard to do basic things that other kids do naturally. But he never gives up, he keeps going, and he tries with all his will to do simple tasks like, you know, crawling and walking and feeding himself, things that we all take for granted. You know, initially it was hard, and it's still hard. Every day is a little harder because Michael has now started to not reach the milestones he should be reaching. He's not walking, he's not talking, and so that affects you internally as a family.
There's nothing. There's no drug. There's no treatment for Michael's specific disease.
When we were told about Michael's diagnosis, they just said, go home and just live your life, but don't expect much from your son. He will be in a wheelchair, he will be nonverbal, he will be delayed, and there's nothing we could do for you.
Doing nothing is impossible. We can't just sit back and say, Michael can fall apart and that's okay. That's not okay for us. You know, we'll do everything possible.
There's a process that could help Michael, but it involves this very expensive approach. Alzheimer's disease, multiple sclerosis, Parkinson's disease, ALS, and I can go on and on. All of these common diseases are genetic diseases that are more complex, meaning more than one gene is involved. To fix those diseases, we need to learn first how to fix the diseases where single genes are defective, such as Michael's disease. It is the research in these rare, terrible diseases of children that is going to be the springboard for us to figure out how to treat the common diseases that afflict every one of us.
His prognosis is his future looks bleak. He can't fight for himself, but that's why we're fighting to do it fast and quick. So we're just hopeful. We're hopeful that we make it. It's hard.
Hey. Hi.
Sorry. After the event and that video that we did, we went viral, and we did galas and golf tournaments and you name it, we did it. We were out there, bike rides, and we were able to continue his raising money, but at the same time, he was making progress, as you see here. He was almost walking, but the disease was also kicking in. And after a 3-hour seizure that Michael had one day, he lost the ability to become verbal. He was verbal for a while, but it hasn't come back since, unfortunately. And then the world locked down, the whole world locked down from COVID. And instead of us worrying about how we're going to raise money or how we're going to make this therapy, we were just worried about not losing any more function for Michael, because we couldn't get any therapists to come up to the house. No one wanted to help us. They weren't allowed to come to the house and do therapy anymore, or for us to go there. So I reached out to our community again, asking for help. And this is what they did a week later after me reaching out saying, I need help for Michael. So at the height of COVID, this amazing team came together, blocked off my street, brought food from my neighbors, went one by one into the backyard, building this pool, and we got Michael in therapy to continue his therapy, so we wouldn't lose the ability or wouldn't get more paralyzed as we continue this journey. So now we had Michael doing therapy, and we were trying to think, okay, well, how do we raise money? I started riding a bike in the evenings to get physically fit during COVID. And my wife was like, well, why don't you ride from Toronto to Vancouver? I'm like, well, hold on. This is 4,000 kilometers. I'm barely doing 10. This is me on the bottom here, dead after 10 kilometers. But I went to my MP and I said, if I happen to ride from Toronto to Ottawa, would you make it so I could meet the Prime Minister? And they thought, yeah, we should be able to. So I started training from 9 till 2 in the morning, 3 days a week, as far as I humanly could. And I went from 10 to 20 to 50 to 100. And then after about 7-8 weeks, my mom and dad drove in front of me for 100 kilometers a day, 4 days in a row. Finally made it there, and I pleaded the Prime Minister at the time to make sure that we got more rare disease funding, not just for Michael, but for all the children. Unfortunately, it fell to deaf ears. He didn't do anything about it, but we raised $250,000 or just under that on the bike ride, and we continued our journey. So now we had Michael in therapy, we had funding, and then thankfully the NIH went in right when they could, and they started testing the cells, and they proved here that if they add the gene therapy to the cells and increase the amount of gene therapy, that we're able to recover the cells. So the cells in red are the ones that weren't recovered, and the ones in green that were. And as you see, as it goes up, we're recovering more cells. Now, it doesn't show any toxicology, it doesn't show anything like that, but at least it showed that the gene therapy was going to work, or we thought it was going to work. And then what happened was we got the mouse data from the wild-type mice showing that at 1 month, 5 months, and 1 year out, that we were increasing expression. So expression was increasing over time. And that, well, it's a good sign saying that the gene therapy looks like it's working in the mice as well. And the big question became, do we now spend all the money and do everything concurrently? Because if we did everything concurrently, we can lose or save around 2 years of work. Instead of losing 2 years, we could gain 2 years. But the problem was that we would have to gamble almost $2 million. And we did that. We said, whatever it's going to take, we'll just do it. So we did a rat a toxicology study, and we got great results in dial distribution. We then did a non-human primate study, and we showed, again, great distribution. All this data is published by Dr. Gray, this type in SPG50 toxicology. He did an excellent job of publishing this data. And then we showed in mice that we could restore handling and clasping. So on the left is what a normal mouse should look like. On the right is a mouse that is affected by SPG50. And we were able to show that the mice then were able to then do the spread eagle, showing that they've recovered from or didn't get the disease. And then with all this data, we're able to then target a human dose of 1e15, which is the highest dose ever given in a human being. And one program did do this before us, which was CLN7, and they didn't see any major toxicity events with the children. And then basically with all the collaborations that we did between Boston Children's, UTSW, the NIH, and a lot of companies that offered their time and support and services from their amazing teams, we received Health Canada approval on December 30th of 2021 after filing a CTA. And then on March 24th of 2022, Michael was treated at Sick Kids Hospital, where this journey began. The hospital was amazing. They supported us. They were there from day one. And we were very blessed to have such an amazing team of people. And I just want to call it out. It wasn't me. It wasn't Dr. Gray. It was a team. A team of people came together to save this child of mine. And they did it out of the kindness of their heart. They did it because they cared, and they really went above and beyond what it really took to get there. And if you look at the timeline, excuse me, I'm blowing my nose here. If you look at the timeline, from the time that Michael was diagnosed, we had nothing, no mice, nothing at all. And from the time that he was diagnosed until the time he was treated was just under 3 years. And the reason why it went so quickly is we had the clinical batch made to treat children before the non-human primate studies were even done. So if this would have failed, we would have lost almost $4.5 million. But we were lucky; everything worked out, and we continued along. And if you look at the total funding that it cost us, the base biology was understood. It was understood for 20 years, so we didn't have to fund any of that work. The gene therapy cost around $500,000 to create and do. It was about $1 million to $1.5 million to do the safety studies, because we had to do rat and non-human primates. Then about $2 million to manufacture the drug. This is actually not zero. Sick Kids invested about $300,000 to treat Michael. And then we had 2 more doses after Michael was treated, and we opened up a phase 1/2 in the US to treat 2 more children. That cost about another $1 million. And this is the teams that we want to just acknowledge and thank. It was a team effort. A lot of amazing people involved, and I couldn't be more blessed to know these people and I've worked with them. And then basically what happened was we went to give our program away, the license, the drugs, everything, and no one wanted it. They basically said it was too small of an indication, that there was no financial benefit for these companies, and we couldn't get it out to anybody. So I decided that I had to do something, so I quit my job. We formed Elpida Therapeutics. We're now a 5-person company in California. I live in Toronto. Everybody's scattered, remote, hybrid. And our goal is to run like a nonprofit and to save as many children as humanly possible with these ultra rare conditions. And if you look at the chart here, and I'll explain it, it's a bit complicated, or it looks complicated, but it's not. We dosed Michael in a phase 1/2 at Sick Kids, and then we opened up a phase 1/2 at University of Texas Southwestern Medical Center. Thanks to Dr. Manassian and Dr. Einacone and Dr. Gray. And we treated 2 children at that hospital for the phase 1/2. We then had a mother call us with a 22-month-old child, explaining how she was pregnant. She called me back 3 weeks later, confirming that her unborn child, who's now born, had the disease, and we decided that we had to do something. So we treated the baby at UTSW, the youngest ever to be treated intrathecally at 5 months old. And then we heard about another child in London who's going to be treated next month at 7-month-old. We then treated a baby last week, or a child last week, in Spain. And we're going to be opening up sites in Spain, Italy, and Germany. And we're going to be treating 12 patients there this year, thanks to Virogen, who is our manufacturer in Spain, who agreed that they would make more drug at no cost as long as we're willing to treat the children, which we were. And then finally, we received FDA approval to do a phase 3 study in the US, and we'll be dosing those patients in August at the NIH, UTSW, and Cedars-Sinai. So overall, we'll be treating roughly around 22 children with this gene therapy this summer into the new year. And then what happened was we were part of the Bespoke Gene Therapy Consortium at the NIH, and they couldn't take on another program because of funding. And thankfully, there's a group called CIRM in California called Center for Innovation, Research, and Medicine, who agreed to partner with us on SPG50 and CMT4J to take them forward. We received a $4 million grant. We finalized toxicology studies last month. We're waiting for the data to be released. And then if everything goes well, we'll be starting a natural history study next month and treating patients early next year. We're waiting for 3 more programs. We're actually in the works of trying to decide which ones they are. But unfortunately, we're just low on funding right now. So we're arranging to have them added on, but until we have funding and we're dedicated to take them to the end, they'll just be on pause. And if you look at the treated children that we had so far, this is Jack. This is the youngest child to ever be treated intrathecally. This is his sister Naomi. Hopefully, she'll be in the study starting in the spring, in the summer. This is Jack from Connecticut, and this is Alberto from Spain. And then we also have 1 more child treated as well, or 2 more children, to make it 5 in total, including Michael. And these are the faces of the children that are waiting. And I think a lot of times we forget about all these amazing children that are waiting for these therapies, and these are the faces of them. And our goal is to treat as many as we can. Hopefully, all of these children on this image here, or on this presentation, will be treated hopefully this year. And I just want to just briefly go over, I think everybody knows this, but if not, I want to go, how big is this problem of rare disease? And I think a lot of people don't realize how big it is. I personally think this is a humanity level problem with so many people involved. I mean, when you talk about there's more than 10,000 rare diseases, more than 400 million people affected worldwide, and more than 95% have no treatment. This is a pandemic. This is worse than COVID, and we need to figure out how we can make this happen, because when there's only 500 of 10,000 diseases that have a treatment of any kind, there's a problem, and we have to try to fix it. And I think the biggest number that kind of shocked me when I heard about it was more than 3,000 children die from rare diseases every single day, and that's just unacceptable. You know, to have that many children dying every day from a rare disease is just unbelievable. Yeah, and I think the biggest thing that when we look at this and you say, you know, as families, what do we go through? Well, you know, this is a bag that sits in my corner of my bedroom, basically getting ready anytime that Michael may have a problem. We have to run to the hospital, and parents shouldn't live through that. Parents shouldn't have to have a bag sitting in the corner, getting ready to go to the hospital any given time. And our children shouldn't go through pain and suffering to understand what disease they have or the disease they have. And they shouldn't live like this. They deserve to live better. And parents should never have to bury their children. It just should never happen, especially for a rare disease that a lot of them can be treated or cured. So I say to everybody that's on here, if you are doing research, if you are a doctor, if you are a parent, something can be done. You can try. It doesn't necessarily mean it'll be a gene therapy. It could be just giving your child better physiotherapy. It could mean doing a drug screen. It could be anything, doing a gene therapy like I did. But if you try and you try to do something to improve your child's life and advocate for them, you can do something. And if you're a researcher out there, if you're a company, I just ask you to work with urgency, because you know a brother, a sister, your grandfather, your grandmother, someone you know will be affected by a rare disease. And if we do something now with urgency, maybe we can save this generation, not just the next generation. And I just want to thank everybody for your time, and I'm open to answer any questions that people may have. Thank you.
Thank you very much, Terry, for sharing the story and the path to N=1 gene therapy. I think it's quite a lesson for many others.
It's a crazy journey, that's for sure. It was blood, sweat, and tears, that's for sure.
Yeah. I don't see any questions in the chat, but maybe I can start just by saying that it looks that the main roadblocks here, of course, one is funding, because we all know that industry doesn't have much interest for very rare indications. So is there any alternative to family based fundraising to make it easier for these diseases to reach a gene therapy approach for this
Yeah,
very...
I think when you look at it, like, I mean, in the end, I was, we were an introverted family. We were very close, to ourselves. And then we had to come out and tell our story. And I think the more people that tell their story, they come together as not just one family. If they come together as a group of families together and they try really hard, they can raise the money. Because in the end, you're not raising $4 million. What you're doing is you're raising pieces of the journey, right? You're basically fundraising for proof of concept. When that shows that there's results, then you can go back and say, now we're fundraising for safety testing. When you're done the safety testing, you're fundraising for manufacturing. When you're done for fundraising for manufacturing, then you're raising to dose to children. And I think if you do it and you break it up into those pieces, they're manageable instead of saying we have to raise $4 million at one point in time. And then obviously the longer that you do these things, the more time you have to raise the funding. Unfortunately, there is no easy answer. It is really about funding, and working with urgency, unfortunately. But things can be done as long as there is a path for a gene therapy. I think the other big thing that is a big roadblock is the safety, right, of these programs, because there's a lot of things that are coming up, like DRG toxicity. There's heart problems that people are getting from high doses of gene therapy. Then there's some brain conditions, inflammation in the brain after gene therapy. So there are some safety concerns. But again, if everybody thinks through the problem properly, thinks through the disease, these can be mitigated. And I think one thing that is really important is to think of ourselves as copycats. If we're in this field and we're doing this, we're not going to create a new capsid. We're not going to, as a family foundation or as a researcher, or we're not going to, come up with something that hasn't been done before, because we don't have the money to go and do 30 non-human primates or 100 non-human primates or a study on 100 people. We have to do a small, nimble, minimal viable product. That's what you would call it in IT, to get to patients to be treated, and then think through the bigger problem is how do we get the drug to everybody.
So I think one of the points you just mentioned is the use of so-called platform technologies. So to use validated gene therapy vectors and approaches that have been already tested for other indications.
Exactly. So like
accelerate.
Yeah, and then when I talk about the gene therapy 101 class, you can go online. I'll make it available. What you do is you try to find the same capsid, right? Yeah. AV9 or AV8 or AV2, the same promoter that's been used before, and the same route of administration, right? If you copy those three things, then your path will be a lot easier, because there'll be data on 1, 2, 5 programs done before, where you won't have to do it. Now, it might not be the most optimized for the disease. It may not be the best out there, but again, it's very difficult for a foundation to develop something from scratch. Obviously, if it's like a Schwann cell or something like that, where AV9 doesn't work well, then you're going to have to do something different. But again, it's something that we just can't copy. We have to copy. We can't create, unfortunately.
Yeah. One more question from me. So can you comment a little bit on the sequence of the development steps in your case, and in general, what can be the most efficient to make sure that we do develop personalized medicines, but we also get the regulatory approvals in terms of, whether the requirements are for a natural history study, and which step you have to involve the regulatory authorities, and what is the optimal path with the least delay for the treatment?
Yeah, it's a good question. I think it depends on how big the disease is, but I'll just talk about a rare and ultra rare condition as an example. So let's say a Michael's disease, right? You could go, so the path to a gene therapy, I'll talk about that, then we'll talk about the natural history. So the path is you do a proof of concept and you prove that the gene therapy works in cells, in mice, maybe organoids. You don't necessarily have to show it in mouse, right? You can show it in just cells, but it has to be a really good cell model, and then they prefer to see a mouse. But let's say that you go and you create 2 mouse models and they don't show any phenotype at all, right? You can go to the regulator and say, look, we tested in cells, we tried in mouse, and either the mouse dies right away or the phenotype doesn't show anything. You can go to the regulators, but they do want to see that you tried. I think that's the most important thing, that you tried. Then I would also say that you run long-term safety data in a wild-type mouse while you're proving that the gene therapy is working. And the reason for that is, if you can show it in long-term mice, it gives them just a little bit of that comfort level to say, hey, you did it in a year's worth of mouse. There was no serious adverse events. And then that's more, again, you're building a story to say, this is safe and it's efficacious. Those are the 2 things you have to definitely show in your story. Once you get past proof of concept, then you manufacture what's called an engineering run to show that the drug can be made in a very small amount. And then you make a toxicology batch. That batch is meant to be dosed in animals. And all this process after the proof of concept has to be the same, so that your toxicology batch and your human batch are done in the same process. So it's like baking a cookie, right? If you change the ingredients or you change the directions, that's not the same cookie anymore. It's a different cookie. The same thing with gene therapy. If you change anything, the product or the directions, it's considered a different product. So you have to follow the same thing. You can't leave companies and go somewhere else. Once you start your engineering run, you're hitched to that company, right? So it's really important that you partner with someone. Well, so what happens is, once you make the toxicology batch, you then do it hopefully in rats. That's the easiest path. You dose, different cohorts of rats. You show on different dose levels, low, medium, high. And what you do is you try to make it so that your human dose is 3 times in the middle, and you go 3 times higher and you go 3 times lower. That's kind of what normally you do. You can go 2 times lower, and 2 times higher, but kind of try to keep it at 3. And the purpose is, you want to kind of show that at 3 times higher there is toxicity a little bit, and that your dose is somewhat safe, because you want to know if there's a limit of where you can get to, and to show that there is actual effect. Once you're done the toxicology, then you go and you make something called plasmid. And you made it earlier, but now it's more expensive. So now you make the plasmid. It's the raw material to make gene therapy, and then you make the batch, your manufacturing batch. And once that's done, then you can dose patients. Now, I never mentioned where the regulator piece fits into there. So once you're done your proof of concept and you show that it works, you then go to the FDA or your regulator for something called a pre-IND meeting. A meeting where you want to file an IND, you want to get the drug to the children, but you want to ask them if you've done all the steps necessary to appease their requirements. And again, usually it's 2: efficacious, but most importantly, that it's safe. And that's it's a scale, because if your disease is fatal, then the scale of safety can kind of go a little bit in either way. But if your disease is non-fatal and your children don't have any kind of outcomes, then the bar has to be set very low for any safety events. So you go to the FDA and you say, here's my package, here's the efficacy, here's the safety, here's everything that we've done. And then they come back and they give you compliments. They say, you know what, you need to show biodistribution in the animals, or you need to do this, and this. They'll give you guidance on what they need to do. You answer those questions over the next 6 months to a year. You manufacture your drug. You do all the tests that they need you to do. And then once you're answered all their questions and you feel comfortable that the safety is there, and you manufactured your drug, you then file an IND. And the IND says we're safe to proceed to dose a patient. The FDA comes back, gives you approval. Then you have to go to the hospital, and you have to go through something called the IRB board. And it's like an ethics board and a protection board to protect the hospital from doing something wrong. You go to the hospital, you file to the IRB, and there's usually maybe 2 or 3 IRB boards depending on the hospital. Once you're done that, then you have to train the staff, you have to do all these documentation, and then you can dose the patient. That's kind of from beginning to end. Where does the natural history fit into this? It's a really important question. If it's a small indication, you may not understand the course of the disease. It may not just be possible. In that case, you'd use something called a phase 1 study, which is just meant for safety. You're not trying to prove efficacy, you're just trying to prove that it's safe. If you have efficacy, a natural history to say, like us, Michael will be paralyzed by the age of 10, by the age of 20, he'll be quadriplegic, he also, seizures, blah, blah. Then you can file for a phase 1/2 study, because now you kind of understand the course of the disease and it kind of has a progression. But if your disease has a wild dispersion of disease, for example, CMT4J, our second program, there's 4 diseases in one. There's people that are affected by 1, they die by the age of 2. People that are affected by the age of 5, they get affected and they pass away by the age of 14. People that are affected by 5, they pass away by the age of 30. And then people that are 30 and get passed away by the age of 50. How are you going to match the controls? How are you going to do the natural history for that? It's very difficult. So if you do have a natural history, then you do a phase 1/2, which is safety and efficacy. Now, let's say that you did a prospective natural history study. You followed patients for 6 years or 5 years. You know the exact course of the disease, and you have some initial safety data. You could follow a phase 1/3 study, show that it's safe, show that it's efficacious, and then show that you can make a change in that patient's life that correlates to something. Now, that could be a placebo control, which we don't want to do in our patient population, or some sort, again, or the natural history study, if it's good enough, to match that patient to say they were supposed to be paralyzed by this point, but they're not. That's kind of, or they're supposed to be, passed away by now, but they're not, as an example. And that's how the process is.
That would be the ideal design for an ultra rare disease, of course. I see. Thank you very much. That was really insightful. I have a question from Dr. Gerbild. How do you think we can engage the clinicians here in Europe? And how is Michael doing today?
I mean, I think it's I think each country is different. I think some countries are easier than others. But I think what you need to do is you need to find someone that is motivated, right? Someone that is I call it like, someone that wants to go to war with you, right? That's what you're doing. You're going to war with that person, right? So if you find that person and they're like, I will help you, I will do everything I humanly can, but you have to respect their time that they're giving you as well. It's a two-way street. That's the person you want. You want someone that is not trying to get you out the door in 15 minutes because you have another patient, someone that's willing to take the time and effort to try to figure out how they can help you. The second one, how Michael's doing pretty good. As you can imagine, even though we called it Cure SPG50, a cure is very difficult to do. And I think what we got to think of is the earlier we get children, the sliding bar towards a cure is in that direction. The later we treat children, the more towards a treatment it is, right? So it's like a sliding bar. Michael was 4 by the time we treated him, unfortunately, because it took us so long, almost 5. So what we see in Michael is we think we stopped the spasticity from expanding from his ankles. But one thing we are seeing, which is drastic, is the cognition improvements, which we never expected. All the children, so far, all the families are saying spasticity seemed to hopefully have stagnated, but the cognition has been improving in all these children. Is that compared to baseline? It's really hard to tell because we're still very early on our clinical trial, but that's what we're seeing in most children is that cognition. Michael now has a tablet. He tells us what he wants to eat, what food he wants to eat at that point in time, what clothes he wants to wear, what shows he wants to watch. He is able to maneuver his wheelchair like no tomorrow. So he's improving. It's just, I wanted to cure my child. I wanted my child to be normal and get married and have kids. And unfortunately, that won't be for Michael. But maybe it'll be for that baby that we treated or the next child, right? That's our goal.
Yeah, thank you. I think that also brings an important aspect of what expectations we as clinicians and researchers and also families should have from the gene therapy, which depends, of course, on the situation of each patient and the stage of the disease.
And I think, I think what happened was we had this amazing gene therapy. We had SMA, right? We had kids that were supposed to be dead by the age of 2, and they're running around by 10 years old. And then we had AADC deficiency, where kids were paralyzed, and you inject into the brain, and you see them going to school 2 years later. But we have to remember that those were engineered diseases. Those are diseases that someone chose. They felt they could change. They're not super complex in the course of the disease. And don't get me wrong, these are all complex. But when you're talking about like a transporter or a lysosomal storage disease, which there's multifaceted things that are happening. I think, in some diseases we're going to cure the kids, and other diseases it's going to be a treatment, right? And I think that the expectation should be just stopping the disease. That's kind of like, that's the expectation for all of them, is let's stop it from getting worse, right?
Yeah. So there is another question. Thank you for a wonderful presentation. It's anonymous. Another thing is that in general, rare diseases take a few years to get diagnosed. Obviously, you had that too. Nearly 1/3 of the rare diseases show the first onset in neonatal period. So prenatal, postnatal testing are extremely important. And I think advocating for screening is equally important. And wishing you the best to little Michael and your family.
Thank you.
So
Yeah, I mean,
that's
one thing we are doing is we are advocating not just for our diseases, but for all diseases. We should have whole exome sequencing for all children that are born. I think that's fundamental. I mean, there are a lot of challenges. Like I've been hearing, for example, some hospitals in the US are doing that for every patient, and then parents are giving up their children after finding out they have a fatal disease. So there are these, or insurance won't cover them anymore. There's some things we need to work out for sure. But I think if we offer it to every child, then we'll be able to diagnose them so much quicker. And especially if the drug is available, either in a clinical trial or available, it'll change their lives. But it's very complicated. It's a very complicated thing to figure out how we're gonna make it so we don't, lose insurance, so parents don't give them up, and how we improve their lives, and also how we don't treat children that may never get the disease, right? Making sure that we're treating the proper children, because we may think they're going to have the disease, but they may not, right? So
yeah, it's the limitations of the screening. Sometimes you don't know the significance and of the variants. There is another question by Linda Lafontaine. Is the information generated through your therapy usable to other universities in order to help other rare diseases?
Yeah, so the toxicology that we did can be used, because what happened was Dr. Gray really showed that between a mouse and a non-human primate, that the DRG toxicity and the biodistribution is very similar. So it hopefully will, if you reference the document, that you won't have to do a non-human primate study. If you also go to elpidatx.com and you go under documents, I actually published all of our protocols, our IB, our 2.4, all the documents you need to go to the FDA, or the majority of the documents. I published those as well, so that everybody can use that as a template, and not have to spend that much time and effort like I did to create those documents. Because when you create those documents to the FDA, you're telling a story, and the story has to be told in a very specific manner. So I published those documents, and you're more than free to use them. And thanks to the collaboration of all those people that you see on that website, we were able to actually release them.
Okay, I mean that's obviously very important. The sharing of the knowledge, and especially for these ultra rare diseases, is really important to be able to transfer know-how, technologies, and information from one disease to the other, because that's the only way to accelerate. Okay. I don't see any more questions from the audience. I think we covered most of the aspects. So I just had one more technical question. So do you have any age criteria for treating the patients, and do you expect any difference when delivering intracerebroventricular or intrathecally, lumbar? What is the difference there?
Yeah, I mean, for example, I'll talk about the two that I know that we've compared, or Dr. Bray has compared, and a few other people, Novartis has compared, cisterna magna and intrathecal, right? Those
Yeah.
two have been compared really well, and I think that one just showed that you're getting more to the lower spine, one's getting more to the inner brain, but there's very similar in disease and very similar in distribution. I think intraventricular is a very interesting one, delivery method as well, and I think it depends on the disease and what you're trying to hit. If you're trying to hit upper motor neurons, then intrathecal works better. If you're trying to hit like the cerebellum or deeper into the brain, then it makes more sense for you to do intraventricular or direct delivery to the brain like you're doing for ADC deficiency. So I think it depends on the disease, what cells you're trying to hit, to determine what route of administration you're going to do.
Okay.
I don't think there is a wrong answer, obviously, depending on what disease it is, but I think there's the opportunities are there, and enough programs have done those different scenarios that nothing will stop you from doing them. You just got to make sure that you choose the right one.
Okay. Great. Thank you very much. I think if there are no more questions, I would like to thank you again for the very interesting, very personal and very inspiring presentation. I think it's an example for many. I hope that it will bring a lot of success, and it will have a real benefit also from Michael and all other kids. And I would like
If anybody has anything they want to talk about, if they have a disease that they're working on, if you're a parent and you want to try to do something, you reach out to me. I'm traveling quite a bit, but if someone needs help, if there's a disease you want to take on, if there's something you want to do, I'm here to help and support you and try to save the child that you're trying to save, or the children. So count me in. I'm in 100% on this. So reach out anytime, and I'm more than happy to help.
Yeah, thank you very much. It's really inspiring for the community. And I would like to thank everybody who was also attending today and for your participation. And also, I would like to remind you all that we have some more webinars coming up. Please don't miss those. We have next week Professor Dimitri Kalman from UCL. He will talk about gene therapy developments for epileptic disorders, so May 23rd. And then we have genetic therapies for muscular dystrophies on May 29th by Dr. Annemieke Artsmarus from Leiden. So very interesting webinars coming up. Stay tuned. Thank you very much. Thank you, Terry, again.
Thank you, everybody. Thanks a lot for your time. And yeah, really good seminars, really good people you're going to have on. So,
yeah, for sure. Thank you. Bye-bye.
thanks. Bye-bye. Bye, everybody.
Thank you. Bye.
Bye, Elizabeth.